With that, I think I can go ahead and introduce the first talk of our session this morning or this afternoon. And this is going to be coming from Dr. Heather Mefford, who is a clinician scientist with training in pediatrics and medical genetics. And she's a member of the faculty at St. Jude Children's Research Hospital in the Center for Pediatric Neurological Disease Research. Her research laboratory works to identify and characterize genetic causes of pediatric epilepsies. And today, Dr. Mefford is going to be discussing the genetics of Dravet syndrome. Remember to drop any questions you have in the Q&A chat because Dr. Mefford will be joining us live after her presentation to answer any questions you have. Thank you all so much. Looking forward to the day listening to these exciting talks together.
Thanks for having me today to speak to you about genetics and Dravet syndrome. Now, on the surface, I think genetics and Dravet syndrome is somewhat simple because for the most part, Dravet syndrome is solved. But what I want to talk about today is to get into a little bit of the nitty-gritty, to talk about what is the gene or genes that cause Dravet syndrome, what are the types of mutations that cause Dravet syndrome, and walk through some of the details that might be helpful when you're interpreting either genetic reports from testing or talking to family members about SCN1A, Dravet syndrome, and mutations. I always like to start as a geneticist by talking about why having a genetic diagnosis matters. This may be obvious to some of you, but I think there are lots of things to think through and good reasons to do genetic testing, even when we know, have a good guess about what the answer is going to be. Having a genetic diagnosis confirmed helps us as physicians talk to families about what is the prognosis for you or for your child, given the genetic diagnosis. We can also talk about recurrence risk or the chance that this will happen to another family member, depending on what the inheritance of a mutation is. And more, especially in the world of epilepsy, understanding the genetic diagnosis helps us decide which medications either to prescribe or to avoid. Importantly, it also provides research opportunities for us to develop new, more precise medications and connects families, as you are all connected here through the Dravet Syndrome Foundation, who have the same genetic diagnosis. And this is important both for supporting affected individuals and their families, but also for supporting researchers and having a collection of individuals with the same diagnosis who can provide information about that diagnosis and their journey. And in the case of foundations like Dravet syndrome, to provide funding to help accelerate research. And of course, our goal is always to use the genetic information to develop, to understand therapeutic targets and develop more precise therapies. So as many of you probably know, Dravet syndrome was first described actually in the late 1970s, but it was described based on its clinical features. And we didn't have the tools at the time to dig into why does Dravet syndrome occur at the genetic level. And the clinical diagnosis is briefly summarized here. This was initially proposed by Charlotte Dravet. And so physicians would have to make the diagnosis of Dravet syndrome by observation of the patient's features. It was in about 2001 that genetics came onto the scene for Dravet syndrome, when a group identified de novo or new mutations or pathogenic variants. So I'll use interchangeably mutation and pathogenic variant, which mean the same thing, in the gene SCN1A as a cause of Dravet syndrome. And this was a really remarkable discovery, showing that this is due to a genetic mutation and not something environmental or otherwise. So let's talk about the inheritance of Dravet syndrome. And it's actually, for most individuals, not technically inherited from either parent because it's due to a de novo or new mutation. The mutation that happens in the child occurs usually either in the sperm or the egg, but prior to fertilization. So then every cell in that embryo and child carries a mutation on one copy of the SCN1A gene. And that's just shown here, where this represents 2 little bits of a chromosome in dad, the same 2 bits in mom, and the child inherits a normal copy from one parent and a copy from the other parent that developed a mutation. And today, when we do genetic testing, we can actually identify changes in the SCN1A gene in about 80 to 90% of kids who present with a clinical diagnosis of Dravet syndrome. I think it's important to think about, people often wonder, well, why does this happen? Why does it always land in SCN1A and cause Dravet syndrome? But if we think about from a genetic perspective and cellular perspective, every time we make a sperm or an egg, we have to copy the entire human genome. So that's 3 billion letters. And so every time an individual is born, they actually carry 50 to 100 de novo or new mutations in their genome. So everyone listening to this talk today has changes in their genome that we wouldn't find in either one of your biological parents. If we look only at the genes, so genes like SCN1A, SCN2A, and all the other 20,000 genes in the genome, every individual has probably between 0 and 5 or 1 to 5 de novo changes that affect a gene. And even when a de novo change happens in a gene, it doesn't always cause a disease. But when it lands in the wrong place in that gene, and we'll talk about the types of mutations those are, that is what leads to disease. So there are many conditions, not just Dravet syndrome, but many often severe childhood developmental conditions that are due to de novo mutations like this, which are really just a consequence of a copying error in the genome. The other type of mutation that you may hear about is also a de novo mutation, but it's a mosaic mutation. And mosaic just means that we are a patchwork of cells. And when someone has a mosaic mutation, it happens only in a subset of the cells in our body. That means that the mutation didn't happen in the sperm or the egg, but that it happened somewhere in a cell as the cells were dividing during embryogenesis. And so that in the child or the individual, either the tissues and organs that are affected, or how many, what % of each cells in each tissue and organ carry that mutation, really depends on how early in development it happened. Importantly, mosaic mutations can be hard to detect by sequencing tests, so they can be missed, and that's something we'll talk about towards the end. But we're getting better and better at sequencing, and so often we pick these up. And certainly they can be disease-causing when they occur in a significant enough fraction of cells. And then finally, I'll mention that you may have also heard of parental or gonadal mosaicism, which means that in the parent, a fraction of the cells of the sperm cells or the egg cells carry an SCN1A mutation. And that parent may be completely unaware and unaffected because it only affects those gonadal cells, or it may be present in a few other cells in the body. But importantly, this may increase the chance of having additional affected children with the next pregnancy. So let's talk about what these mutations are. What do I mean by mutation? So in order to talk about the mutations, I'd like to take a step back and do a little genetics 101 so that we can understand how we detect and interpret those genetic changes. So I like to think of our genome, or all of our DNA, which remember carries all the instructions for building our cells, our organs, and a functioning individual, as a set of encyclopedias. Our DNA is packaged into chromosomes, which you can think of each of these chromosomes as an individual volume in the encyclopedia. So each volume carries slightly different information. And within those volumes, or scattered along the chromosomes, are the genes, which are the actual instruction units. And so if we zoom into a paragraph in that one of those volumes of the encyclopedia, we see these red sentences that we can read and understand that carry instructions. Between those sentences is a lot more sequence, which here I've indicated is really unreadable. We can actually interpret some of it, but it's slightly different than reading the sentences and the instructions. We have about 20,000 genes, and these make up only about 1% of our DNA. But interestingly, we know how to read these genes. There's actual code that turns the DNA into a protein. And so we can read that and detect errors and predict what sort of problems those errors are going to cause in the protein. So here's an example of what we would see as a spelling error, right? If we change one letter in this sentence from a D to an F, we actually change the meaning, right? From wash the dishes to wash the fishes. If we change a different letter, so from the to thy, so an E to a Y, it actually doesn't change the meaning very much. And so this is still understandable and may not cause a significant problem. It's these types of spelling changes that cause many different human diseases, including sickle cell mutation, which I've shown the actual mutation here. But the majority of Dravet syndrome is caused by these type of changes as well. There are other types of changes which actually are more deleterious. So here we change the E to a stop or a period, and now we can't read the sentence. So it doesn't work. And this is the equivalent of what we would call a nonsense stop mutation that prematurely truncates the protein or causes a shorter protein. And actually, most of the time, those shortened proteins don't actually get made. And then finally, we can talk about splicing mutations. And this is where you connect two parts of the gene that were not meant to be connected. So here we've changed the sentence to gibberish, and this also does not make a functional protein. This would be a splicing error or can sometimes be caused by other types of changes as well. So how do we find these spelling errors? Well, we sequence the gene. And we used to do this one gene at a time. So if we suspected Dravet syndrome in a patient, we could sequence the SCN1A gene and look for mutations. And if we didn't find them, often we were done. Today, we have better tools, so next generation sequencing or high throughput sequencing. And in most cases, we don't just sequence SCN1A, we sequence a large number of genes that might cause seizures and developmental delay in a child who's affected, or we sequence all 20,000 genes, which is the whole exome. But the result is the same. We identify spelling changes that we then have to interpret and say, is that genetic change causing disease? We can do this several ways. So we use a lot of different information saying, have we ever seen this genetic change in healthy individuals? If so, it's probably not causing Dravet syndrome. We can use those predictions, so reading the sentence or the gene to determine whether it's going to actually cause damage to the protein. And we also look at inheritance. Is it a new change in the affected child, or is it inherited from a parent? Now with SCN1A, there are individuals or families who have varying severity of epilepsy. So in rare cases, the SCN1A mutation is inherited, but there we take in all the information about whether the parent has symptoms as well. This is a cartoon of the SCN1A protein. So it encodes a sodium channel, as you probably know, and this is showing you just a piece of a cell, the outside of a cell. So this is the membrane around the cell. And SCN1A is a transmembrane protein, or the protein actually just snakes in and out of the cell. And in 3D, it actually creates a channel that allows sodium to go back and forth from outside to inside or inside to outside the cell. And so I show this because it's important, because changes in different parts of the protein may have different effects on how the protein works. So the most severe types of mutations I already talked about briefly are the stop mutations. And these actually cause just a shorter protein. So the cell is reading along to make the protein and is told to stop making that protein, but it's too soon. So these shortened proteins actually often don't get made at all. The cell has ways to detect that this is abnormal type of the protein and just destroys it. So the result is that there's not enough SCN1A protein around for the cell to function properly. The other types of mutations we talked about are the missense or spelling errors, right? So here is an example where in the middle of this section of the protein here, we change one amino acid to a different amino acid. And the result of that may be that it disrupts how the protein folds or how it lets sodium in and out of the cell, and actually changes how the protein works. And again, the end result is that there's not enough normal functioning SCN1A protein in the cell. These are a little bit more difficult to interpret because we all have some differences in the actual sequence of our proteins. But we use all that other information about whether it's been seen before, predict how it might affect the function of the protein, and sometimes even test that to determine whether it's causing disease. And in some cases, there may be more mild or more severe effects depending on what the actual change is, as I showed you in this example before. This is a schematic from a review by Dr. Catterall several years ago that represents all of the changes that have been seen in the SCN1A protein to date. These are the missense or spelling errors, each of these dots. And the stars represent places where there's a stop mutation in the SCN1A protein. And the red stars and dots are the ones that cause Dravet syndrome. So what's important here is just that there are many different mutations in SCN1A that can cause Dravet syndrome. They're spread throughout the protein. But you can see there are some places where perhaps missense changes are more likely to have an effect. And many of those occur in these regions that cross the cell membrane. And there are other regions where having a stop seems to have a significant effect, though the stop mutations almost always cause disease. So a question that often comes up is, can we predict how severe disease is going to be, or what type of epilepsy my child will have if we know what the mutation is? And the short answer to this question is not really. There are individuals, even in some families, where there's a parent with GEFS plus or a milder form of epilepsy whose child has the exact same mutation and has Dravet syndrome. So there's a lot of work to try and understand this, both in Dravet syndrome and in many other genetic diseases where there's a range of severity. The hand-waving answer is that the genetic background is probably important. This means, what are the other changes that you have in your genome that make you different from your parent or from another patient with Dravet syndrome who has the same mutation? And we just don't have these answers yet. We need a lot more patients to study and a lot more information. That said, there are rare examples, and I'm just showing one here that we published last year with Dr. Lynette Sadlier from New Zealand, where a change in SCN1A actually causes something that looks different than Dravet syndrome, but it's always the same change. So in this case, if we see that change at amino acid 226, we can predict with some certainty what that patient will look like. So finally, what if there's a clinical diagnosis of Dravet syndrome, but they can't find a change in SCN1A? So for example, if your child's neurologist says this is Dravet syndrome, but you've had genetic testing and don't find a mutation, what does that mean? Well, there's been work to look for other causes of Dravet syndrome, so mutations in other genes that cause Dravet syndrome. And there have been some suggestions of this, including one that we published here several years ago, where mutations in other genes cause something that looks like Dravet. But for the most part, and this I think is an interesting description here, for the most part, although many genes have been described as Dravet, causing Dravet-like syndrome, most of them kind of have their own flavor. So they look a little bit different from Dravet syndrome. So it's possible that it's not the true classic Dravet syndrome that we think about. The other possibility is that we're missing the mutations for different reasons. So this is showing you one of those instructions. So think of this as the paragraph, and the exons are the sentences that we can, actually the words of the sentence we can read. And what happens in the cell is that these words or exons get spliced together and cut out part of the DNA so that we can actually read this fluidly. And that is where what the protein gets made from. And when we do genetic testing, we usually sequence just the parts that we can understand and read, the parts that get spliced. So that's the exons and a few little attached bases on either side. So these are the regions that get sequenced. But we know now that there are mutations in other parts of the gene that can cause disease. That includes in these intervening or intron regions, the promoter region, which actually helps turn the gene on and off. But they're rarer and they're harder to interpret, so we don't always look for them when we do clinical testing. That said, there's a lot of effort to find these types of mutations. And I'll just give you one quick example of one that we identified a couple of years ago. And here I'm just showing you, say, 2 exons of SCN1A. With a little bit in the middle here that is not part of the normal SCN1A protein. So usually these 2 exons get spliced together and the cell makes a normal, full-length, functioning SCN1A protein. But when this purple bit in the middle gets spliced in and included, it introduces a stop mutation or a stop instruction. And that causes the protein to be shorter and actually just not made in the cell, similar to what I talked about before. So there's not enough SCN1A protein, or there's less, sorry. And this is actually normal. It happens normally in cells maybe where you don't want SCN1A expressed, or at a developmental time point when you don't want SCN1A expressed. So the cell knows when to include this and when not to. But we identified some mutations or changes in and near this purple exon here that actually cause the cell to include this poison exon, and we call it poison because it causes the protein not to be made properly, to include this poison exon more frequently. And so if you're making more of this non-functional form of the gene compared to the wild type form of the gene or the normal, then the result is basically the same as if you have a stop mutation in SCN1A. You have less normal protein, and that causes Dravet syndrome or GEFS plus phenotypes. And we were able to show this in a small number of families. So it's mutations like these that we don't normally look for that might be causing that last 10% or so of patients where we can't find the mutation. And I'll just give you a preview. You're going to hear about this later, but this same poison exon, or it's also referred to as an NMD exon, is actually being targeted to try and treat Dravet syndrome. So that's just a teaser, and you'll hear more about that in another talk. And then finally, there are other types of mutations. I told you that this region of the gene actually helps turn the gene on and off when it's supposed to be on or off. And there are ways that, and this happens by these proteins or blobs here sitting down and saying, turn on SCN1A in this cell. But there are other types of changes to the DNA, which this one is called methylation, that can actually prevent those proteins from sitting down and turning the gene on. And again, if you don't turn on the gene and make the protein, you won't have enough around, and that can cause Dravet syndrome as well. So we're looking for these types of changes in SCN1A and other epilepsy genes in patients where we can't find mutations that cause their disease. So I'll just summarize here your Genetics 101 talk. I hope it's been helpful. We know that mutations in SCN1A cause nearly all cases of Dravet syndrome, and we're still looking for those other mutations that we might have been missing. They can be missense or truncating variants. They can happen throughout the protein. We talked a little bit about how that happens and why that's important. And then research is ongoing to find additional types of disease-causing variants, and of course, to target SCN1A loss so that we can improve and even cure disease in affected individuals. So I'd be happy to take questions about any parts of this talk. It's been a pleasure talking to you. Thank you.
Thank you so much, Dr. Mefford, for that talk. We're going to go into a live Q&A. I have a few questions filtering in from the chat so far. So the first question that came in, it asks if it's not genetic, and I think maybe they meant actually if it's not inherited, can the parent have another child with the same disorder?
Right. So if we've identified a mutation in SCN1A that looks like a new mutation in the affected child, the risk is low that that the parents would have a second affected child. However, I did mention early in the talk that parents can have a few either sperm cells or egg cells, depending on the parent, that might carry that SCN1A mutation. And that happens maybe 5% of the time, where there's a parent who might have some extra germ cells that carry the mutation. And then there is a risk that they would have a second child. It's difficult to detect and the risk is low, but it's certainly not zero. So it's a tough question, and it's one we deal with a lot in the genetics clinic.
I think the next question, in the context of this being a genetic disorder and a disruption of the SCN1A gene, can individuals outgrow Dravet syndrome?
So the mutation in the SCN1A gene is usually present in all of the cells of the affected individual. So they have one working copy or normal copy of SCN1A, and then one that carries the mutation. And that will be present throughout life. So I think the short answer is no, though everyone kind of develops, who has Dravet syndrome kind of develops at their own pace and has their own trajectory, though you can get a sense for what that's going to look like, from a clinical picture of other kids with Dravet syndrome. So no, you don't outgrow it, and the mutation does not go away over time.
I'm getting several questions coming into the chat asking basically, what are the tests to be able to detect mosaicism? And, should all parents be seeking out, tests to try to detect mosaicism if they're interested in having more children?
That's a great question. So we can do there are some tests we can do. None of them are perfect, and none of them will give you an answer that says there's absolutely zero risk that you'll have another affected child. We just don't have that ability. We can test the parents. We can test their blood, which is what we usually do with the kids. We can test their saliva, because just kind of two different tissues, and see if there's any chance that we can just see a hint of the mutation that the child carries in the parent's DNA. And sometimes we can. So sometimes we can detect the mutation in the parent's blood or saliva or skin. And then we can say, okay, we see it, so we know that there's a risk with the next pregnancy that the child will be affected. It's usually less than 50%. So in a genetic disorder that's passed on from grandparent to parent to child, when it's passed on in that way, the risk with every pregnancy is 50%. With mosaicism, it's usually quite a bit less than that. I think whether or not you pursue that testing, in part, depends on why you want that information and what you would do with a subsequent pregnancy. Do you want to test the pregnancy for the mutation? You could always do that regardless of whether the parent, we know the parent's mosaic or not. So that's always an option. So just thinking about how you would use that information to inform family planning or what you might do with a subsequent affected pregnancy.
What about, if parents, what if their blood was never tested? What if their genetics were never looked at, and, we're 10 plus years later, maybe they're not interested in having more children, but is there another reason that they should pursue more genetic testing? Is there a reason that knowing if it's de novo would make a difference?
That's an interesting question. I think some parents who are mosaic may have the mutation in enough of their cells that they might have had febrile seizures as a child, or might have had some seizures during their lifetime. But that doesn't really change, I think, with the genetic testing. And if they're not affected, and if none of the siblings are affected, I think it's highly unlikely that anyone else in the family carries the mutation that's disease causing. So probably not. I think it's worth looking at the extended family as well, to see, is there anything in the family that might suggest the SCN1A mutation is in other individuals, and that might be a reason to test, but that would be unusual to be completely unaffected.
And so then another follow-up on this same topic. So what about siblings? So if you have an unaffected sibling of a child with Dravet syndrome and they're not presenting with symptoms, should they still be tested, especially, thinking about would they have any chance of passing this on in future?
Usually in that case, we would test the child if they had any hint of carrying the mutation. So for example, if they had febrile seizures or if they had an unexplained seizure sometime in childhood, then it would probably be worth testing. If they're completely unaffected, I think it's something that when they become an adult and are thinking about having their own children, that they may want that information to consider whether or not they want to be tested. But again, if you're completely unaffected, it's much less likely that you carry the mutation.
Okay. I just got a question coming in, and I think we got a similar question yesterday, and so I really want to get this in. And it's something I see parents trying to wrap their head around a lot, is what could possibly be causing these de novo mutations? Are these just random, or were parents exposed to something during their lifetime that caused these mutations?
Great question. These are random. So, as I said early on, I think these are spelling errors. And if you think about, again, that encyclopedia analogy, and if you had to go in and copy every sentence, every paragraph, every volume, let's say you're typing, you would make typos, right? And this is what happens in the cell. Remarkably, there are, with 3 billion base pairs, 100 typos is actually not very many. So the cell does a really, really good job of copying every piece of DNA almost perfectly, but it's not perfect. And it tends to be fairly random where these changes land in any given cell. And so it's not related to anything you ate or drank before or during the pregnancy. It's not related to exposures. This is something, like I said, that every one of us, on the planet carries de novo mutations. It's just that when one of them lands in SCN1A to change the protein, that's when you end up with Dravet syndrome. And I should say that this happens in many other genetic conditions. So it's not just Dravet. Many of the other severe early onset epilepsies, the mutations are de novo. Many genetic syndromes that we tend to see in genetics clinic, they often occur de novo. And there are other examples as well.
Great. Another question I'm getting a few of is just asking, is a germline mutation the same thing as mosaicism, or is it indicative of mosaicism?
So we often use the term parental mosaicism, germline mosaicism, gonadal mosaicism to mean basically the same thing. That just refers to the fact that there's a mutation in usually a small fraction of germ cells, which are the sperm or the egg. Somatic mosaicism, when we say somatic, we mean body tissue, so it could be any tissue. And so some kids with Dravet, like I said, have mosaic mutations, and those we refer to usually as somatic mutations because they occur in the tissues throughout the body, and they occurred during development. So yeah, there's a lot of terms that mean almost the same thing. Yeah.
But yeah, so a child having mosaicism is not indicative of a parent passing on mosaicism. It's de novo, right? So it came new during the development. Okay.
Yeah, if a child has a mosaic mutation, that's about the only time we can say there's almost zero chance that the next pregnancy would be affected, because it didn't happen in the sperm or the egg, it happened in that individual.
Great. So I think one more question, and it kind of harkens back to another question we were discussing, which was sort of that idea of de novo mutations being random. And I think in the context of the way you presented it, our cells have a really great spell checker to make sure we're not making those mistakes, but some things slip through. And so I'm just getting another question asking about, environmental factors, that parents are worrying about maybe that led to more mutations. So this was specifically asking about a cell phone tower, but I have a lot of parents asking about, my own vaccines that I got as a child or an adult or things like that. And so do you really feel like most of these are random, or do you think there are some environmental factors that could be impacting, or both?
That's a tough question. There's no good evidence that that most environmental exposures or factors have any influence at all on the mutation. There are rare examples, right? So very high exposure to radiation, for example, for individuals who are in Hiroshima, right? But that's not most of us. And it may be over the next 100 years that we start to figure out what the very subtle effects are of some environmental exposures. But for the most part, when we look across populations and we look at how many de novo mutations do even normal individuals have, it's really consistent across a population and does not suggest that there are significant environmental factors. That's not to say there couldn't be things we haven't yet detected yet, but then the effect is probably quite small because we haven't been able to find it.
Okay, that makes sense. Thank you so much for joining us for this Q&A. Thank you for your really informative talk. And
thanks for having me.
Everyone can join me in a round of applause and thanking you. And with that, I think we will move to our next speaker.
Great. Thank you so much.
Thanks.